Wonder how my cells grow. They divide their nucleus. And mitosis is divided.
Welcome to another virtual biology lecture. Today we're going to be discussing cell cycle and mitosis. Hope you guys enjoy and don't forget to take good notes. Today's virtual lecture's objective is to become familiar with the following assessment standards from reporting category 1 that are going to be tested in the STAR Biology EOC. At the end of the lecture, you should be able to explain the cell cycle as well as the disruptions that may take place in the cell cycle leading to certain diseases such as cancer. Now, in previous lectures, we've mentioned single-celled organisms as well as multicellular organisms, and despite how different they might seem, they do still have some characteristics that they have in common, such as the ability to grow. Now, unicellular organisms grow as new matter is added to their one cell. Multicellular organisms, on the other hand, they grow as the number of cells making up the entire organism starts to increase. For these organisms to grow, however, it's important to note that cells must be able to reproduce or make more cells like themselves, which brings us to cell division. Cell division is the process through which the cell divides to form two new cells called daughter cells. Now, there's three basic types of cell division. The first type of cell division is in prokaryotes, which are unicellular organisms such as bacteria. The second type of cell division is in eukaryotes, and in this cell division, the purpose is for growth and repair. Now, the third type of cell division is also in eukaryotes, but the purpose of this one is to produce sex cells, also known as gametes, which we're going to learn more about in later lectures. Now, for this lecture, we're going to be focusing on the first two types of cell division. First, let's talk about cell division in prokaryotes. Now, in prokaryotic cells, cell division is relatively simple. Prokaryotes reproduce through binary fission. The first step is for the cell's genetic information, DNA, to be copied. We're going to talk about DNA copying in our next lectures, but once this DNA has been copied, the cell then divides into two, with each new cell receiving a copy of the genetic information. The two daughter cells are identical to the parent cell. The second type of cell division is seen in eukaryotic cells when growth or repair is needed. Now, this type of cell division is a major component of the cell cycle. The cell cycle basically describes a eukaryotic cell's entire lifetime events, both during growth as well as division. The growth stage is known as interphase, which is the longest stage, and the division stage is known as the M phase. But before we take a closer look at what happens during the cell cycle, let's recall that eukaryotic cells consist of a nucleus, which directs all activities of the cell and stores chromatin, which is the organism's genetic information. It's extremely important to note that chromatin is long spaghetti-like structures that are made up of DNA, deoxyribonucleic acid, which is actually what contains genetic information. Now, if the cell were to simply divide like that, it would only get half of its genetic information, which is why it's important for it to first make a copy of all of its genetic information before it can actually divide. That way, when it divides, each daughter cell will receive a complete copy of the genetic information. The copying of genetic information occurs during the biggest component of the cell cycle called interphase. Now, interphase consists of three phases, G1, S, and G2 phase. The first is the G1 phase, also known as the first growth phase. During this phase, the cell undergoes rapid growth, and it carries out all of the processes needed for life, such as homeostasis, energy conservation, transport of materials, and synthesis of new molecules. Basically, this is the stage where the cell is just growing, so it's actually increasing in size. The second is the S phase, also known as synthesis phase. During this phase, the copying of genetic information begins to occur through a process known as DNA replication, which we're going to discuss in greater detail in our next lecture. The third phase is known as the second growth phase, or G2 phase. During this phase, the cell prepares for mitosis. But what does this mean actually? Well, it's actually the shortest phase, and it basically just prepares the nucleus so that whenever it is going to divide, and makes additional organelles and cytoplasm so that the cell has enough to divide in two. The second component of the cell cycle is actually where the cell divides, and it is known as the M phase, which consists of two stages, mitosis and cytokinesis. Let's start with mitosis, which is the process responsible for dividing the nuclei. Nuclei is plural for nucleus, so this phase basically is responsible for each new cell to receive a copy of every single chromosome. In humans, that means that each new cell will receive 46 chromosomes. Once mitosis is complete, the cell divides into two daughter cells that are identical to the parent. This is actually the process known as cytokinesis, which is where the cytoplasm and the organelles are divided into two cells. Let's take a much closer look at mitosis, which has its own stages, 4 in total, known as PMAT. P for prophase. Now, this stage is responsible for chromatid condensing. Basically, it tightens up into what we know as chromosomes. Remember, chromatin is made up of DNA, which is why we say chromosomes carry genetic information. Now, you may be familiar with a chromosome's look, such as this X shape, but that's not exactly an accurate description of all chromosomes. Yes, this is a chromosome, but half of that X, like this, is also considered a chromosome. Chromosomes don't get the X shape until their DNA is duplicated, in which the complete set of genetic information that is tightly packed in one chromosome is doubled, like this, forming an X shape. For instance, humans have 46 chromosomes in every single nucleus of every single cell in their entire bodies. When the genetic information is copied, we still have 46 chromosomes, but now we have 92 chromatids, 2 copies of each chromosome. Now, we still refer to it as a chromosome because it's the same information, just doubled. But now we refer to each copy in that chromosome, in other words, each side, as a chromatid. This is where sister chromatids join at the centromere, which is the center, forming X shapes that we often recognize as chromosomes. But once these chromatids get separated at the end of the cell division, in which one sister chromatid is given to one daughter cell and the other is given to the other daughter cell, they are now called daughter chromosomes. Notice that they are not in the X shape anymore, and still they're considered chromosomes. So keep in mind that that X shape just means that the genetic information has doubled. During prophase, the nuclear membrane also breaks down and the centrioles, which are found in the cytoplasm, separate and travel to opposite poles of the cell. This causes spindle fibers to surround the centrioles and attach themselves to the centromeres of each chromosome. Next, we have M for metaphase, which is where the nuclear membrane has completely broken down and the chromosomes have lined up along the middle of the cell with spindle fibers attached to each chromosome at their centromeres. Then comes A for anaphase, which is where the sister chromatids from each chromosome are pulled apart by spindle fibers and begin to move to opposite poles of the cell. T for telophase, which is where chromosomes reach opposite poles of the cell and begin to unravel once again into a huge bundle of chromatin as it once was. The spindle fibers also break down and the nuclear membrane reforms around the chromosomes at each pole. At this point, the cell has 2 nuclei, each with a complete set of genetically identical chromosomes. Once mitosis is complete, cytokinesis follows by actually dividing the cell along with its contents of cytoplasm and organelles, thus in the end giving you 2 identical daughter cells. The diagram above summarizes the cell cycle, which I like to remember as IPMAT-C. I for interphase, which is where the cell spends most of its lifetime in. PMAT, which corresponds to mitosis, starting with prophase, metaphase, anaphase, and telophase. Then lastly, you have C for cytokinesis, when the cells finally divide. As we mentioned at the beginning of the lecture, the new cells produced during cell division enable the organism to grow in size by adding cells. However, not all new cells are used for growth. In fact, many new cells are used to replace old, dying, or damaged cells. For instance, skin cells are short-lived. Typically, they live about 2 to 3 weeks, which is why your body needs to keep replacing those cells, or let's say that you cut yourself, those damaged cells must be replaced by new cells as well. Also, there are some cells that no longer want to divide, or maybe they don't need to do so at the time. They tend to enter a phase known as G0 phase, which is a phase in the cell cycle where the cells exist in a resting state, meaning they are not dividing. Now, that doesn't mean that they're not doing anything. They're still doing their regular cell functions, but they're just not dividing. The G0 phase is sometimes viewed as an extended G1 phase because the cells can enter this phase for a while, and then down the line when they need to, they can re-enter the cell cycle into the G1 phase so that they can prepare to divide. Most multicellular eukaryotes are made up of many kinds of specialized cells. For example, your body has skin cells, muscle cells, red blood cells, liver cells, and many others. Now, these different types of cells are given their unique assignments early on in the organism's development through a process known as differentiations. Now that you know that new eukaryotic cells form through mitosis, it's important to note that most of these specialized cells in a multicellular organism divide to make other cells like themselves. For example, a muscle cell will divide to produce two new muscle cells, while a liver cell will divide to produce new liver cells. In most cases, the new cells that are formed through mitosis are identical to their parent cells. However, sometimes the cell cycle is disrupted. Disruptions in the cell cycle can lead to diseases such as cancer. Cancer affects the mechanisms of cell division, so either abnormal cells or an unusually large number of cells are formed. In some cases, these cells join together into a large mass known as a tumor. The cell cycle can be disrupted when genetic information is not copied correctly before the cell divides. Now, the body has ways of detecting and correcting many of these errors, but those mechanisms are not perfect. Environmental factors, including radiation, tobacco smoke, and other pollutants, can also change cells and disrupt the cell cycle. You're going to learn more about these changes in genetic information of cells that can affect the organism in later lectures. But now, I just want you to make sure that you understand that eukaryotic cell cycle and its purpose, which is for growth and repair. Okay, now that we've gone over the cell cycle, as well as the disruptions that may take place in the cell cycle leading to certain diseases such as cancer, you should be familiar with the following assessment standards from reporting category 1. This concludes our virtual biology lecture on cell cycle and mitosis. I hope you all took great notes and wrote down any questions or concerns that you may have so that we can discuss in class. Well, that's it. Have a great one.
Wonder how my cells grow.